guys! Our article is called Hydrophobic Residue in E. coli Thioredoxin Critical for the Processivity of T7 DNA Polymerase. This article was trying to figure out what interactions at the interface of thioredoxin and DNA polymerase promote high processivity. Therefore, the experiment was intended to test human thioredoxin, which is similar in structure with E. coli thioredoxin, to see which structure caused difference in the results if the results were different. You might be wondering what is this girl saying, but don't worry, we'll explain to you these terms. Keywords to explain here are thioredoxin, processivity, and DNA polymerase. So first, thioredoxin. Thioredoxins are proteins that facilitate the reduction of other proteins through cysteine-thiol disulfide exchange. The thioredoxin kind of looks like this. And there are two cysteines here. These two cysteine at the end are major key to the capability of thioredoxin to reduce other proteins. This is the structure of cysteine, and this is the major part where the reduction happens. This is the better demonstration of thioredoxin than my crappy drawing. So before going into how thioredoxin performs reduction process with its substrate, let's briefly recap what reduction process is. Remember oil rig? Oxidation is loss, reduction is gain. So simply, reduction is gain of electrons and oxidation is loss of electrons. As you can see from the picture, A is giving off an electron to B. So atom A, oxidation is taking place. Or you can say atom A is being oxidized. On the other hand, B is gaining an electron, which is reduction is taking place. Or you can say atom B is being reduced. Should be N. So this is the reduction process of thioredoxin. As you can see, cysteine in thioredoxin attacks disulfide bond of the substrate. Cysteine of the substrate gets reduced and substrate and thioredoxin forms disulfide bridge. As soon as this happens, cysteine 35, another cysteine residue of thioredoxin, forms disulfide bond with cysteine 32, transferring 2 electrons to the substrate, which is now in reduced form. Oxidized thioredoxin is then reduced by thioredoxin reductase, which in turn is reduced by NADPH. Second term, processivity. Increase in processivity simply means DNA polymerase can add more nucleotides for DNA replication with increased efficiency. And last word, DNA polymerases are enzymes that create DNA molecules by assembling nucleotides, the building blocks of DNA. These enzymes are essential to DNA replication and usually work in pairs to create 2 identical DNA strands from one original DNA molecule. As I said before, thioredoxin works with DNA polymerase to speed up DNA replication process. Before looking at how thioredoxin works with DNA polymerase to speed up DNA replication process, let's quickly recap basic concept of DNA replication. Replication is the process by which a double-stranded DNA is replicated to produce 2 identical DNA molecules, and it occurs in 3 major steps: the opening of double helix, the priming of the template strand, and the assembly of the new DNA segment by DNA polymerase. So first, initiator protein unwinds DNA double helix. Then the helicase attaches and breaks apart the hydrogen bonds between the bases on the DNA strand, which fully pulls them apart into two strands. Meanwhile, Primus attaches to each strand and assembles a foundation. Then DNA polymerase can now wrap itself around the strand and attach new nucleotides to assemble new complementary strand. Like couples during cuffing season, A is paired with T and C is paired with G. And there is no couple swapping. Again, this is a better picture than my questionable drawing of DNA replication process. You can stop the video to look through it, but we will move on. So basically, thioredoxin forms a complex with DNA polymerase to increase the processivity of nucleotide polymerization. Here we have GP5, Gene5, DNA polymerase, and TRX working together for high processivity. Isn't this so cool? Thank you for watching, and please click like and subscribe down below if you have enjoyed the video. Bye!